Hey everybody. So this chapter on cell membranes is kind of big and very important because the membrane is that boundary between the outside world and life. All life begins at the cellular level. Cells are living things. And this membrane is semi-permeable, meaning it keeps some things out, it lets other things in. It's loaded with proteins that have different functions, as you've already read about, and it can communicate. It's involved in communication with other cells. So signals have to get in and out of that membrane so we can have a multicellular organism like ourselves. Here's an example, once again, of transport using proteins in the membrane. In this case, we have receptor-mediated endocytosis. And we have a normal cell here, which is loaded with proteins in the cell membrane that bind to cholesterol. So when you eat food, cholesterol ends up in your bloodstream after digestion. And these proteins bind to cholesterol and bring it into the cell. And that's typical. And that cholesterol can be used to build more cellular components, or it could be digested. Now, in some of us, we have fewer of these proteins, and we end up having extra cholesterol in our bloodstream. And for some of us, we don't have any of these proteins, and we end up with a lot of cholesterol in the bloodstream, and that leads to disease like high cholesterol, right? We have, and that could lead to plaque forming in our arteries and stroke or heart attack. This is a common phenomenon. So if you mess with that membrane, things don't get in or out. Just like if you don't have the right chemical signals, you can't get sugar into your blood as easily, or into your cells as easily, and you end up with diabetes. So when things are in the blood and they can't get into the cell, it's usually because of a missing protein. Now, cells, as I said, are living, and you're composed of trillions of them. But you are not just a pond full of cells, you're a multicellular being. That means your cells are in tight communication with one another. And the way these cells typically communicate are through chemical signals, much like hormones, right? We do also have a nervous system with electrical signals, but even that has some chemical components, as we'll see. So cells can communicate in direct contact with one another. Cells in direct contact communicate through gap junctions. They send signals through gaps in their cell membrane. But those that are a little bit further apart have to rely on other kinds of signals. And those that are on one end of the body than the other need a totally different kind of communication. So we're going to look at local and long distance signaling. Of course, direct contact is one of them. Direct contact is one way they'll talk to each other. But if they're not in direct contact, we have something called paracrine signaling, which you can see here. Paracrine signaling is when cells are not in direct contact with one another. They are side by side or close together. They share the same interstitial fluid, which is that fluid around the cells. But what they do is through exocytosis, they produce, so let's say they produce a protein in the rough ER. It then is put into a transport vesicle, sent to the Golgi apparatus, where it then goes to the plasma membrane and is exported through exocytosis, where it goes to a nearby target cell and causes some kind of response. That's paracrine signaling, signals sent to nearby cells. Okay, so we could have cells in direct contact, or we could have cells nearby that communicate through paracrine signaling. Another kind of local signaling is synaptic signaling, and this is part of how your nerves work, your neurons. But the way this works is you have one nerve here and another one here, and they almost touch, and that little gap between them is called the synapse. So let's say your brain tells your leg to move. Well, that electrical signal is sent down your brain, but wherever one neuron ends and another begins, a chemical signal is sent through exocytosis. These are called neurotransmitters. So they diffuse out of the synapse and they hit the next cell, and then they allow that signal to continue. So one type of local signaling is synaptic signaling. Now, other kinds of chemical signaling in your body are long distance. So here we have hormonal signaling. It works a lot like paracrine signaling. See, we have exocytosis from a cell, in this case in the brain, hormones being released in the brain, but it goes into the bloodstream and then it is transported throughout the body. So this is long distance signaling or hormonal signaling. The fancy word is endocrine signaling, right? That means hormonal. So endocrine signaling is a long distance kind of signaling where signals are put into the bloodstream and sent throughout the body instead of just local cells. Now, here's what's going to happen next. We have a signal molecule, and this molecule is usually referred to as a ligand, which is a word you might not know. I don't have a pen with me, so let me see if I can. It's a ligand, ligand. Much like the word ligament, a ligand is one molecule that binds to another. Okay. And so this signal is a ligand. It binds to a receptor. This is a protein in the cell membrane. This is the step of communication known as reception, when a ligand binds to a receptor. When that occurs, then it causes a change inside the cell. This signal does not come into the cell. It's polar and large. It won't cross the membrane. So other molecules in the cell are activated, and that's called transduction. That's how the signal is sent throughout the cell. And that leads finally to response. And that response could be a number of things. In the example here, it says the activation of glycogen phosphorylation, which is a molecule that adds a phosphate group to a glucose molecule, making glucose more reactive. That's something we do all the time. So that's something that could happen. There's lots of things that could happen based on this response. Your cell could be told to divide. Your cell could be told to self-destruct. Your cell could be told to bring in glucose. If this molecule was insulin, it would bind to an insulin receptor, and it would lead to sugar being brought into the cell. Okay. Of course, if you're diabetic, you may not produce the signal, or you may not have the receptor for the signal, and that could lead to diabetes. All right, so the three steps of communication, once again, are reception, transduction, and response. Now let's look at a more specific example of this, a specific kind of reception. This is a kind of receptor known as a G protein coupled receptor. Okay, so this molecule here is the G protein coupled receptor, and see this circular thing here? And this thing over here, let's just look at this. This thing right here, this is part of your G protein. Okay? And what happens is the ligand docks to the receptor, the GPCR, the G protein coupled receptor. It then adds GTP, which is kind of like ATP, but it's guanine instead of adenine. It adds guanine TP triphosphate to this G protein, which then activates it. So a lot of molecules, a lot of proteins get activated when phosphate is added to them, phosphate groups. Phosphate groups have a lot of energy. When they're added to this molecule, it causes the protein to change its conformation, which means changes its shape, and it basically gets turned on to its active shape. Once that happens, that activated G protein goes on to activate an enzyme over here. It docks and it activates this enzyme, and that leads to a response. So this is reception, this is transduction, leading to a response. Now we're going to look at a specific example of this. This is how, for example, your fight or flight reaction might kick on with adrenaline. It can be activated this way. So an adrenaline molecule is sent, docks to a liver cell, G protein receptor fires up the G protein, which is this oval here. The G protein triggers a response, and that response would be the release of glycogen into your bloodstream, so you have a lot of energy to run away from a grizzly bear or something. Another example of a type of receptor is an ion channel receptor, where a signal docks, and instead of something like activating a phosphate group or something, instead what happens here is the channel opens and it allows diffusion to take place, and then it shuts back off again. So this is a way to turn diffusion on and off. You open a channel, you close a channel with the help of a receptor protein there. Now, not all chemical signals stay outside the cell. Some of them come into the cell. The ones that come into the cell are your typical hormones, those that are made up of lipids. They're non-polar. So non-polar substances can dissolve in the plasma membrane and cross into the cytosol. And in the case of non-polar molecules, they have receptors inside the cytosol. So here's the receptor. In this case, we activate this molecule. It goes on and it's going to attach to a gene. And so what it does is it turns on the production of messenger RNA, which produces a protein. So this is how you might produce a protein. Like, let's say you need to produce, I don't know, new membrane proteins, or maybe you need to produce a growth factor. Maybe the body's been damaged and it's released a hormone that tells your body to reproduce. So the hormone comes into the cell, docks to messenger RNA, turns on a protein signal. That protein signal now leads the cell to begin mitosis. Okay, but the important thing to note is in some cases, in the case of hormones that are not polar, their receptors are inside the cell. So here we have reception, and then transduction is the sending of the signal within the cell, and then the response is the production of a new protein. So let's talk about transduction real quick. Transduction is important because it sends the message throughout the cell, and it can amplify the message. What I mean by that is one molecule can be activated in the cell. That molecule can activate two. Those two can activate four. Those four can activate eight, so on and so forth, and enhance and expand that signal. So here we have an example of what we call a phosphorylation cascade. This is how most of your transduction pathways work. So a signal binds to a ligand, or I'm sorry, a ligand binds to a receptor, right? The signal molecule, or so some kind of signal, some kind of protein docks to another protein. That causes a molecule to become activated. Typically, they become activated by adding ATP or GTP. These are molecules that have a lot of phosphates, that have a lot of energy, and they activate these proteins. And then, so what happens here, notice we have inactive protein kinase 1. It gets activated by this relay molecule and becomes active protein kinase 1. What in the heck is a kinase? That's something you can look up, but a kinase is a molecule that adds phosphate groups to another molecule. So what happens here is a phosphorylation cascade, meaning one molecule adds phosphate groups to another, and then that one adds phosphate groups to other molecules, and those add phosphate groups to other molecules. So this is how you can send a signal within a cell and amplify a signal in a cell. And this is all part of the transduction part of signaling. So this molecule gets activated by the signal molecule. I mean by the receptor after it docks with the signal molecule. This is activated. It activates protein kinase 1, which activates protein kinase 2, and then that leads to activating a protein, which leads to the cellular response. And the important thing to note here is this signal might have been amplified. It may have led to a bunch of proteins being activated, leading to lots of cellular responses. And what I want to show you here, one thing I really want to point out on this diagram is it shows these glowing proteins. That means those proteins have been turned on. They've been activated. And notice that this activated protein has a phosphate group stuck on it. It's been phosphorylated. It's been activated by a kinase. And that leads to the response. Whatever the cell is going to do can now occur. And once again, one signal up here can lead to two molecules being activated, and then each one of those two can activate more, which can activate more, which can activate more, which can activate a whole bunch more. And so as you go down, more and more and more things can be activated, leading to a bunch of response, thanks to phosphorylation cascades. So a cascade's like a waterfall, right? One thing turns on the next, turns on the next, turns on the next, turns on the next. That's what they're trying to get across here with this term phosphorylation cascade. So here's a real world example then of a G protein coupled receptor. These are really important to study. If you're going to study medicine, you're going to learn a lot about these, because this is one of the places that medicine actually works, is on these receptors. Someone just got the Nobel Prize for one of these about 8 years ago for studying G protein receptors. So there's still a lot of research going on how these work. So just to show you, and I'm going to introduce a couple of new words here. Here is a G protein coupled receptor. Here is a signaling molecule. In this case, it's epinephrine. you might know it as adrenaline. Okay, so you see a grizzly bear. Your brain releases epinephrine, like your pituitary gland releases epinephrine, and now what's going to happen is, let me start that over. You see a grizzly bear, your brain sends a signal, your adrenal glands fire up. They release epinephrine. Okay, epinephrine is adrenaline. That gets into your bloodstream and travels throughout your body. That epinephrine docks to a G protein coupled receptor in a cell, maybe in your liver or maybe on your heart. It's going to cause your heart to start beating. It might cause your liver to release glycogen. So how does that happen? Well, first of all, one signal like epinephrine can have lots of can cause lots of activity in your body because different cells have different receptors. They might have different transduction pathways. So they can all receive the same signal but have different responses because of that, because they have different receptors or different transduction pathways. Now, in this example, we have epinephrine docking to a cell, and then that activates a G protein, which then goes to an enzyme called adenylyl cyclase, and it fires up adenylyl cyclase. Adenylyl cyclase then takes ATP. We're getting into more signals here. ATP is adenosine triphosphate. What happens here is it tears off two of the phosphate groups, making it AMP, adenosine monophosphate, and that forms a ring, a loop, which is cyclic AMP. And that is a second messenger, something we haven't mentioned yet. So this is a first messenger, the first one that docks. This is a second messenger inside the cell. That then activates protein kinase A, which leads to responses. Maybe it leads to your heart beating faster, or it leads to sugar being released into your bloodstream. But this all happened because you saw a response, stress hormones were released, your adrenal glands by your kidneys released adrenaline. It went throughout your body, it docked to cells all over the body, leading to different transduction pathways, leading to various cellular responses. And now your heart rate is up, now your body is full of glucose because your glycogen has been released, and you're ready to run or fight. And the term I introduced in this image is cyclic AMP, which is a second messenger, it's kind of like the first messenger that was sent from outside the body, but this is a messenger sent inside the cell that turns on the transduction pathway, which begins at number 5 on this figure. One more example. Here's a zoomed out version here. We have a signal attaching to a receptor that leads to a transduction pathway, a phosphorylation cascade. Notice the glowing molecules indicate they've been activated. So this one activates this one, which activates this one. And then that's going to lead to a molecule that's activated that goes into the nucleus in this case. It activates this transcription factor, which gets turned on and docks to DNA, which produces messenger RNA, which then leads to the production of protein. And that protein is going to cause this cell to divide by mitosis or grow. Okay? So there's a lot of steps here. This reminds me of the child's game if you've ever played Mousetrap. One thing turns on 5 other things, which turns on something else, which leads to the final response. So the things that are important for you to note about all this stuff is I'd like you to know that G protein coupled receptors activate G proteins with GTP. I'd like you to know what a phosphorylation cascade is, what a transduction pathway is. It's a phosphorylation cascade where one molecule activates one or more other molecules until we get to a response. Those responses vary dramatically depending on what example we're using. Now, if you go ahead and you take animal physiology later, which you should do if you're going to be a doctor, you're going to learn about these in much more detail. All right, so this is how our bodies send signals from one cell to another. This is why your heart rate goes up when you have a deer run in front of you when you're driving down the road, right? This is why we're able to run from a grizzly bear faster than we can normally run. It's these hormonal signals being sent from our body. Not only that, this is how we develop. This is why estrogen causes different development than testosterone. It's because these signals cause our cells to grow and develop in different ways. And so a very, very important idea that we're just introducing here in Bio 1.